here and convert it into ammonia. They don't have to just go through a reduction of the nitrate and nitrite that occurs in the soil. Now that process is called nitrogen fixation, the ability to convert the atmosphere into ammonia. Now that's only in the bacteria system, all right? We don't have that, partly because we don't have that enzyme. So here is just a chart to show you what processes can take place. So you can see lightning itself can convert it to nitrate, nitrite, and then of course, depending on the bacteria, they can further convert it. And of course, bacteria also have the enzymes to directly convert through nitrogen fixation, and eventually these all gets into plant, which goes up the food chain to earth. All right? Now, because I'm only dealing with system here that we have processes, I will not cover the part where nitrogen fixation is. So the slide on the nitrogen fixation, those enzymes and pathways, how that is converted, you don't have to cover that. Just know that, we can't do it, but bacteria can do it, all right? Just so that you know. But I won't cover the pathway that the bacteria does, how the bacteria does that. I want to spend the time on Earth. Now, let's talk about essential, non-essential again. So you remember the terminology for essential, non-essential for fatty acids, right? The same applies. When you say essential, it means it's essential to have it in the diet because you do not process the pathway to making it. If you say non-essential, it doesn't mean they're useless. It just means that you're okay if you didn't have it in your diet because you are able to make it yourself. Same logic, all right? And do you remember what are the two essential fatty acids that we cover? Anybody has a memory of that? What are the two fatty acids? I'm asking you, since we are on the topic of essential. What are the two? Don't you want to know it so that you can have it in your diet? What are they called? Huh?
Omega-3 and omega-6.
I can't hear you. Can you say it?
Omega-3 and omega-6.
He said omega-3 and
yes, omega-3 and omega what? 6. But what is the biochemistry name or the scientific name for that? You're right, it's omega-3 and omega-6 that we need as our essential fatty acids. But what is it called when I was teaching you the class? You wouldn't see omega-3 and omega-6 written in your test question, though. Because that's more a layman, the marketing, but you would see the chemical name being used here is different. What would be the chemical name here for the same product or the same compound? Do you remember? How about linoleic acid and linolenic acid? The 2C18 with double bond, one of them with double bond and one of them with the triple bond. Not triple bond. One of them with two double bonds, sorry. One of them with two double bonds and one of them with three double bonds, okay? So you need to know that also for your own health too, right? I know, you go to the market, you tend to be more influenced by the omega-3 and omega-6. So now we also have something like that in the sense that in the amino acids, there are certain amino acids that are considered essential because we do not have the pathway, or somehow in evolution we've lost the pathway of making them, and then there's some that it's okay, you can do without it in your diet because we have the pathway for it. Now just to go back a little bit, why are we studying amino acids? Because they're very important. earlier we already addressed some of these functions. You know, you always want to know why you're studying this in biochemistry. And of course, amino acids is good for what? Making protein. That's very direct and very obvious. But amino acids also contains the nitrogen group or nitrogen atom that you can use to make nitrogenous compound. And there are several nitrogenous compounds in your system. One of the main one, if you remember, is that your DNA is made up of nucleotide. And one of the components in a nucleotide is your base. Isn't your base the adenine, guanine, cytosine, uracil, and thymine contains nitrogen on their structure? So you do need them for that, even that as a basic as your DNA and RNA would require having that compound. And of course that we will go through and you see that there's other nitrogen containing compound that has to be made in your cell. And of course from the intake of food you get your amino acid, and then that amino acids not only goes towards repairing and replacing other proteins, they also replace other components that has nitrogen group in it. Alright? Now, what about an amino acid? We always are concerned with, I think we've addressed this over and over again, by now you'll be tired of it, that an amino acid acids not only have the amino group, but it also have these, which we call the alpha keto acid. And I told you this is not a throwaway stuff. Oh, these goes towards making nitrogenous compound. Well, this is carbon structure. The carbon structure can become intermediate in pathways that can go towards making carbon-containing molecule, or they can be further enters as intermediate to be broken down. So you can see this going into either glycolysis, can't spell today, or TCA cycle, which can be broken down to provide energy. So this can also eventually end up with energy. Or building material. You see either way. So they're all useful parts. So that's why it makes sense to study them. You either use them as carbon source or you can use them to produce more energy in the end. Alright? And now let's look at the essential, non-essential again. I want to, before we go into that back again, I want to talk about some of the terminology and the things that you will encounter. So when we take in proteins and we digest it and absorb the amino acids, they go into a pool in the cell called the amino acid pool. And that pool is where you can then tap to make compounds from it, whatever you want to use it, or if you want to break it down further. Now, if you took an old copy of a protein, that is also broken back down to that pool. So that pool is where you can pool to build, or you can drain that when you break down. And then eventually the very old copy will be pulled out of that pool to be further broken down, okay? So everything goes into that pool. Now the way we balance the nitrogen is, a balanced one means that the intake and output, we have to destroy some of the things. We don't keep old copies for very long. So when the things are broken down, Then there's an excreted part. You can excrete as urea. I will show you how your nitrogen waste is excreted as urea. So if you balance the intake and the output, then there's a balance in it. But sometimes your input can be more than what you break down and excrete, then you have a positive nitrogen balance. On the other hand, sometimes you can take in less, but your output or your breakdown, muscle wasting and all those can occur much more than if you are replacing them. Then you have a negative balance, basically. All right, now let's talk about essential, non-essential. This is a table that already summarizes what is essential and what is not essential. So from the table, it is easy to say that we obviously don't have the pathway for this, for making this. That's why we list them already as an essential component in your diet, essential amino acids. And then of course the ones that we can make, we list them here. Those are the non-essential ones. Whether you have it in your diet is not a problem because you can always make them. Now, then you realize that I take a few things and star a few things and you're wondering why. You already categorize that. Well, I just want you to focus on certain things. Nothing is black and white, straight, easy like that. I mean, yeah, in healthy individuals, this is the format. Then I want you to know that this one, the ones I pick, the isoleucine, leucine, and valine, if you already remember back to your chart on your amino acids, those are referred to as branch chain. And you can look at the structure. The side group has a branch structure to it. That's how they refer to this as a branch chain. Now, the thing that I want to bring to your attention is the stars on these two, arginine and histidine. And why are they starred? Well, in adult and normal individual, those are where they should be. We have the pathway to making it. Although we just barely made enough of it, especially in histidine, we just barely made enough. But in infant and sick children, those are considered to be essential. So although we've listed these are for healthy normal adult, but in infants and sick children, those has to be provided in the diet. Do you understand? So they don't fall under the same category. In their case, arginine and histidine would have to be provided, would have to be supplemented, all right, for healthy living. Now, the other thing that I also double star, which the textbook didn't do, is tyrosine. It's placed here as non-essential. It gives you the impression that we know how to make it. Well, yes and no, and the reason for this is that the pathway to making tryptophan, phenylalanine, and tyrosine actually comes from a common origin. They are together. They branch off, but they share a common origin. And somewhere along the line, so they share a common origin into making, tyrosine, phenylalanine, and then tryptophan, short form. They share a common origin, but somewhere along the line, we've lost all of this. Do you see that? We lost all this. So this is becoming essential, right? We lost that, so essential. But what we kept was this one pathway that I want to show you. So in the past we have that all connected to a common origin. And then there's also one step here, connecting that if you have the phenylalanine, you can always convert that to tyrosine. But over the years, we've lost all of this, but we maintain that one enzyme stack. So that's why I put a double star there. So it's wrong to say that we can make tyrosine all from beginning. We didn't. If you have phenylalanine in your diet, then because we still maintain that one enzyme stack, then we can convert the phenylalanine to tyrosine. So that's what that meaning is. That we consider tyrosine as non-essential. It's only non-essential provided you have phenylalanine. If you didn't have phenylalanine, then where are you going to make it from? Nothing. Because it's the assumption that it can convert from there. Do you see that? So that has to be true. Now, you're going to use back a lot of the things that I've taught you before. Have you remember the term transamination? Transamination is to transfer. It's done by a transferase enzyme. So remember what a transferase enzyme does. You start off with an amino acid. And then you can also have another molecule you can also have another molecule that will then accept that whatever that you're transferring into. So I'm going to transfer this to here. That's a transferase. That's also transamination because we started off with an amino acid. And the amino group is being transferred. So we call transamination. So what happens when this one is transferred to here? Then it's lost that group. So this amino acid now becomes, maybe I should use different color. This amino acid now becomes this group. Let me put this as a prime, just to differentiate. And now this molecule becomes this. Do you see? So they switch. This amino acid is now this, an alpha keto acid. Sorry, it's this. This is the alpha keto acid. When it loses the amino group, it becomes the alpha keto acid. This used to be an alpha keto acid. When it gains an amino group, it becomes this new amino acid. You get that? This process is called transamination. Now, we can add some name to it, and you should help me. What if I add the name what do you think would be an appropriate name for this? Why don't I put it as pyruvate, something that's very common that you already know. So if pyruvate receives an amino group, what do you think pyruvate will become?
Alpha ketoglutarate.
Huh?
Alpha ketoglutarate.
No, pyruvate doesn't sound like alpha ketoglutarate. This one,
Would be
if this one loses an amino group, what does it become? What did you say?
alpha ketoglutarate.
Yeah, that one becomes alpha ketoglutarate. Now think harder. What happens to pyruvate when it gets an amino group? Yes, very good. See, it's not something new. It's something you already know from your glucose alanine cycle. That was what was happening. So I was already teaching you this. So, you might feel, oh, nitrogen metabolism is new. It's not new. We've been talking about this even subtly all the time, even as far back as when we were doing enzyme. And I was talking about transferase enzyme. So transferase is to transfer the entire group to a different molecule. And the receiving molecule is always an alpha keto acid, which is a part of to become an amino acid. It's already the carbon skeleton of a going to be amino acid. Once it gets the amino group, then it becomes the amino acid. And the one that loses the amino group now becomes back to the carbon skeleton, which is whatever carbon skeleton of that amino acid. And so that's always a pairing. The carbon skeleton, and I've already told you about this. Pyruvate is the carbon skeleton of alanine. And glutamate is the glutamate carbon skeleton will be alpha ketoglutarate. Do you see? One more. What is the other pairing that I've talked about? Which way should I say it? Oxaloacetate. You're more in tune to that. That's a carbon skeleton of what amino acid? Which amino acid do you think it is?
Aspartate.
Good. Aspartate or aspartic acid. You see? So that's how, when you learn, now you already get those pairing, and you can interchange. So those pairing works really well in transamination, because one of them will be the receiving, and the other will be the one that becomes the alpha skeleton, alpha keto acid. So you can always pair them really nicely. So those processes are called transamination. The enzyme is also a transferase enzyme or transaminase, just to follow with the same term. Now, this enzyme requires a coenzyme, and that's where your pyridoxal 5-phosphate come in. I've introduced it as a coenzyme very earlier on, but at that time I don't think it matters that much, so it probably didn't go in. But now in this chapter, yeah, it will be very important, right? Because in order to do transamination, you have to make sure you have the right vitamin to be able to make enough of these coenzymes to participate in this reaction, basically. So you see the pairing is very necessary to remember and all that. And then you can pair these together. Each one is going to, then this pair is going to pair with this, because there's always an exchange. Because this becomes this, and then of course, then this becomes this. So there's always a pairing to exchange. So at no point did they ever drop the amino group. They're always handed to something Now, why is that? Why not just drop it? You know, yeah, why wait for pyruvate? Why pyruvate is busy? Why not just drop it and just dump it on the ground or in your blood? What happens? If you do that too much, you know we have 20 amino acids and all of them are lazy in waiting for the other pair, and they all dump the amino group out. What happens? You're going to have a lot of ammonia in the system, and that's not healthy for you. So that's why they always have to wait and say, hey, are you ready to accept it? Because we can't just simply knock it off. It's always capped, handed, so that you don't see a lot of ammonia spike in your blood, kill you basically. But at some point they do. There are some reactions that will allow them to drop it. But most of these 20, when they do the breakdown, they're waiting to hand over that amino group. So it's always safe for your system. So you understand that each pair will pair with the other one. They can pair anyway. Usually, this is the best. Everyone wants to pair with them because this is available in the system quite a bit. So you can take this system and pair it with it. You can take, no, did I lose it? Okay. You can take that and pair with this. Do you see that? And that's where you have your glucose alanine cycle. You can take this and pair with this. So a lot of system likes to pair with this pair in the exchange of the amino group. Sometimes this transamination can also happen basically in this case. This is between, you see this one is slightly different. It's actually because this one can accept another, can have two amino groups. Do you see that? This one has one amino group here and one amino group here. So because of that, then you can have these directly transferred to an aspartic acid to make the asparagine. Do you see that? So this one's a little bit different, where you don't need an alpha keto acid because this one itself has the extra amino groups down the side chain, and then when it transfers from one side chain to another, you ended up with this one. When this one loses an amino group from the side chain, it becomes glutamate, and this one becomes asparagine. Now, this is one of the few rare occasions where you don't need an alpha keto acid to do the transamination. Most of the time, it's always an alpha keto acid be receiving, and then the amino acid become the new alpha keto acid. So I give you the main type, most of the thing, and then on rare occasion you have that, and then this one is a specialized scenario. Happens only at certain location, and it only happens in the liver, all right? And it's very active. Remember I told you most of the time they are transferring, even down to that rare one where you have two amino acids transferring between the side chain. There's always a transfer because we don't want to drop the amino acid, the amino group, which end up forming ammonia. So this is the common scenario. This is another scenario that can happen, but only in specialized location. It doesn't happen all over, all right? Why? Because you're dealing with ammonia now. In this case, you can see if you're starting from glutamate, then the ammonia is plucked out, all right? And this happens. It can happen, but only in the liver. Why? Because the liver is always the detox center. So it can handle, it should be able to handle, otherwise, yeah, then we're in big trouble, right? So in the liver, this specialized reaction can happen because it can handle the ammonia. It also happens that this glutamate dehydrogenase in the liver is very active. So if it comes in this direction, it means that your blood pH is actually low. And it's using ammonia to help bring it up a little bit. As soon as this spikes too much and your blood pH is already okay, it goes in the other direction. That's why it's a two direction. So it's watching. It's not just saying, oh, I just randomly want to get rid of ammonia. It doesn't do that. It's actually a balancing act. So it's monitoring your blood pH, and it's using this item to pH your blood, and if it's enough, you will just incorporate it. So it's going back and forth very nicely, and it's managed really well. This enzyme is highly active in the liver. This enzyme is also not fussy. It will use either NADPH or NAD. Remember, most enzymes only use one coenzyme. This is the first time I ever told you of an enzyme that says, hey, whatever, NADPH, okay, use. NAD, use. You know, even if you guess for exam, you should get at least some of it right. Do you see? Because of this enzyme, it can do both. It can use either one. So it's not fussy like others. So that's why it can do it in either direction, and it can either incorporate that ammonia. If it's building too much in the liver, it will be quickly incorporated into alpha ketoglutarate to make glutamate. On the other hand, if it's lacking in the pH is dropping, then it will move in this direction to do it. So it's really freely reversible, and the excess can shift back and forth immediately. All right? So that's one specialized reaction called reductive amination of your alpha keto acid, basically. It's called reductive amination because we're looking at it from this angle. If I'm looking at it from this angle, then it's reductive because it's using the hydrogen to go in to reduce. And it's amination because it's adding amine group to it. Alright? So that's why it's called reductive amination. I'm going in the direction of incorporating any excess ammonia in your blood into the system. And you can do it the other direction too. Now, that is in the liver, right? Now, what about other cells? How do we deal with the spike in ammonia? There's bound to be ammonia in the system. Muscles break down, sometimes do create. If you don't break down by transamination properly, and you accidentally have some ammonia spike in your system, how does the other cells deal with it? Because I said that one only works in the liver because that's the only enzyme, and that enzyme's in the liver. You can say, oh, can I borrow it? Because I got a little bit of a spike in the brain. You know, it doesn't work that way. You can't borrow enzyme. They are organ specific. So let's look at the brain. The brain really can't stand ammonia. It's very sensitive. In fact, the liver can tolerate ammonia built up much more than the brain. You will go into convulsion and hallucination, delirious and all those, if you even have a spike in the ammonia in the brain. So it's very, very important that the brain doesn't get to that scenario. So what do you have? You have a very active enzyme again, obviously. And this enzyme is called the glutamine synthase? Glutamine. Not glutamate, glutamine. The one with the side chain that has an amino group. Alright? So, and it can be made from glutamate, a close relative, let's call it. You see how the different structure is? This one is an amino acid that is the acid. This is the acid, the side chain with a carboxyl group. This is the amino acid that is the polar one. The side chain is different. Notice the difference, alright? And this enzyme here, glutamine synthase, is very active in the brain. Why? Because it can take free ammonia and incorporate it directly. No more transamination, whatever that is happened to be spiking in the brain, it's quickly incorporated into glutamine. So the brain has that. Now, this enzyme is also present every cell, in other cells as well. But the brain is highly active. It's really working very hard. And I think my notes is wrong. I put a double arrow, change that. You see how I try to highlight it back? It's not, because it serves no purpose if it's going backwards. You know, the brain is never going to say, oh, can I have a little bit of ammonia? I feel like having a pH up. That's never a case. So you know that it doesn't make sense. I was copying paste and then forgot to change the arrow in that. So it has to be in one direction. All right? Now, there is another enzyme that will take it. It's not the same enzyme. Glutaminase will take it the reverse, but it's not in the brain. That enzyme is somewhere else. We don't want to mess with it. So if it goes in this direction, we're happy. Stay that way. And then this goes in the blood to be taken to the liver. Remember? The detox center. That's the whole idea, right? You have ammonia, and you certainly don't want ammonia in the brain. It's quickly packaged as glutamine, safe to put into the blood, and then quickly ship it to the that is the liver. And at the liver, then the reverse can happen. Because the liver needs to take the ammonia out and repackage it, and then put it into the mitochondria. All right? So it doesn't take this into the mitochondria, unfortunately. So you know, it's like every time you ship something to a new factory, they always repackage stuff. So that you have to think of it that way. So this one's in the liver, this one's in the brain, and all cells. All right? So now that you know all the basic part, then we want to address the other part. So you know all the little things that goes on, and now I want to address the synthesis part. How are these amino acids made? Because I told you there are pathways in your system that can make some of your essential, can make some of your non-essential amino acids. So then I have to show you the pathway to making those. Now, learning pathway for amino acid is different from learning pathway for fatty acids and carbohydrates. Those were, to me, very easy because they were, other than gluconeogenesis, they seem to be very direct. Like you have a starting, you have a bunch of, reactions and regulatory steps that you need to understand. You need to have ATP or NAD, H, or NADPH, depending on which pathway we're talking, and then you end with the end product. Same with the breakdown. It's always very much like a straight line flow, not much of a branch or a weave around thing. It's very straightforward. And that's because we only have so few, right? When we're talking about carbohydrate, there's always just glucose breaking down, or acetyl-CoA breaking down, and then when building up, that the gluconeogenesis is the one that you have multiple sources. But in the building of 20 amino acids, imagine how difficult is that? Now you have 20 amino acids to study their pathways, right? If each of them were different. But lucky for us, they were not all that different. Some of the pathways originate from a common source, and that's where they try to consolidate that to make learning easier. So we then divide them because they originate about the same and then they branch off. So that's how you would learn it. You find the common source. You connect those that are made from a common source together. And they do it for you, so that's good. There are 6 families. That means out of the 20, we divide them into families. And why are they in the family? Because they share a common source, the common origin. All right? So here you can see that we're going to study the 6 family, and this is the way you would study it, so that you know. So the glutamate family, and you get introduced to the family, obviously glutamate has to be a member. Otherwise, why would you name it glutamate family, right? So you can obviously say that, oh, glutamate should be a member. And guess who? Because remember how easily we convert from glutamate to glutamine? So that's a family member too. And then of course, now it gets a little bit difficult because now you have to include proline, and ornithine is a non-standard, you come across it again. And then arginine as part of the family members. So now you know the family member. And you should know grandpa. That's the common origin. And you have to know him. Because why? Because it all came through him. Do you understand? So for that glutamate family, that is your common origin. So that's how you would learn it, all right? So this is one pathway where the member shares the common origin. That's why they're all members of that pathway. And that's your glutamate family for you. Now let's go through, it's always important to know why do I have to know this family. And of course, you need to know this family. They are common precursor. Why? Because glutamate plays a very important role in your cell. What was glutamate? Glutamate, of course, is a component, it's an amino acid that goes towards making protein. But glutamate also makes other things. Remember GABA? G-A-B-A, capital G-A-B-A, gamma aminobutyric acid. What do you think that is? Huh? What is it?
I can't remember.
Yes, it's a neurotransmitter. So, glutamate goes towards making your neurotransmitter. Not only it helps make protein, it makes neurotransmitter. And actually, it is a neurotransmitter. It doesn't even have to make GABA. It can also act like a neurotransmitter. It's an excitatory neurotransmitter. So even a single amino acid, you say, it's only one of those glutamate. Yeah, but it plays a role in making things for you. So it can be, by itself, it is a neurotransmitter. It goes towards making GABA, which is a neurotransmitter. And of course, it can also make, and then glutamine. Remember, it goes to making glutamine that helps takes your ammonia away from your brain to be then shipped to the liver. Now, what about glutamine? Other than the fact that it transports ammonia for you from all the peripheral cells to the liver, glutamine is also useful in making your pyrimidine bases as well as your amino sugar. Your amino sugar is like the N-acetyl glucosamine. Those are your amino sugars. And where are they useful? They are in your connective tissue. So here, you haven't studied yet, but glutamine, later you'll see, is to make your nitrogenous base, your purine and pyrimidine nitrogenous base, which is your nucleotides. You remember? So now you can see why, why bother to learn about amino acids? They're only going to make protein. No, because they make neurotransmitter too. They make your nucleotide bases too. So, and you need to know that for test too. Now, what about serine family? Let's look at members of the serine family. It's always, of course, serine has to be members of this family. Who else is member of this family? And you find that glycine is, and cysteine is part of that family member. You see that? And where is the common origin? Glycerol 3-phosphate. I bet you don't remember where you've heard of glycerol 3-phosphate. Have you heard of glycerol 3-phosphate? It's not glycerol 3-phosphate, but glycerate 3-phosphate. Where do you think you have come across glycerate 3-phosphate? Huh?
Glycolysis.
Yes, glycolysis. So when you break down glucose, I won't go through all the steps. After the split, there will be one step here that you would encounter glycerol. So if you want to check it out, it's right after this, after the step or two after the break, the split. You have the glycerate 3-phosphate. So now you understand why I told you what you learned for glycolysis you shouldn't forget, because now you have a place where you know they didn't come from nowhere. It came from glucose metabolism, and that can become an amino acid. Who would have guessed that, you always think, oh, I made protein out of amino acid. Really? If you think harder, where did the protein, the amino acid came from if you didn't take it in your diet? It came from glucose. Do you see? So you never have figured that out. You always think, oh, I made my protein from amino acid. And where did that amino acid came from? If you didn't have it in your diet, where did it come from? It came from glucose. And glucose eventually was metabolized to, form energy as well as some building blocks that you can start to make your serine, glycine, and cysteine in there. So what else did I need to do? Okay. What is the purpose of serine? I think I skipped that step, or I might have mentioned it, but it's not that important then. Remember when we were making our phospholipids? I have to go back that way. When you're making your membrane lipids, I said that you can attach a choline group here. That was what I focused on because I wanted you to know the phosphatidylacetylcholine, phosphatidylcholine as your one of the main component in your. But I also said that you can attach other charge groups here, and ethanolamine is one of those charge groups that you can attach to. And ethanolamine is one of those groups that can be made from serine. All right? And so, and amino acids can also contribute to lipid, your phospholipid. All right? So that's what I'm trying to tell you. Now you're seeing that everything is connected somehow to one another. That the lipids are not just lipid, they could have originated. Their origin can go all the way to an amino acid. the sphingomyelin in your the sphingosine structure that is in your sphingomyelin, another membrane lipids, that comes from serine. Okay, we just didn't do the steps to show you those are upper level, but then I'm just telling you now that those didn't come from, it just made from the structure of a serine can go towards making your phospholipids, basically. Glycine being mentioned, that's the smallest amino acid, but it plays a lot of role. It's a neurotransmitter. Now it's an inhibitory neurotransmitter as opposed to glutamate. All right, so it is a neurotransmitter. That small thing is useful too. And of course, eventually you will learn that it also contributes to making your nitrogenous base, your adenine, your guanine bases will have glycine in it. All right, now I didn't mention this, but these, when you combine it with succinyl-CoA from your TCA cycle, you can make porphyrin, which is your heme group for your hemoglobin. So it's a transport of oxygen. So you can see from an amino acid to building other component. Glycine is together with cysteine. Remember when we make glutathione? The best compound ever in the cell, can have multiple functions, but the most famous function that I introduced to you about glutathione is that it acts as an antioxidant. It reduces the H2O2 so that it doesn't damage your protein and your DNA in the cell. And that's made from glycine combined with cysteine. That's where the SH group comes from. So now you understand why we need to study amino acids and their breakdown and their make. Now the aspartate family actually has a very big membership. Look, so many, but I don't know if it's fortunately or unfortunately, but we lost all these part of the pathway. We only maintain this part. But the plants, lucky for us, plants have all these. So if you eat the plant, you get all that from them. So basically, the aspartate, can go towards making asparagine. All right? Aspartate and asparagine are easily connected. Remember that amino group can be contributed from glutamine, and then or you can start with, and then you end up with asparagine. So they can, here, you see that? A glutamine can provide an amino group to become glutamate, and then aspartate becomes asparagine. And then that's how they are synthesized through a common origin. That's their common origin, oxaloacetate. Not anybody that it's a compound that you're already familiar with. It's part of your TCA cycle, which means then your glucose breakdown again can contribute towards building these two amino acids in your system. Anything important about that? Well, you haven't come across a lot about aspartate, but I will mention it again when we do the urea cycle. And I think I'll leave all those to the urea cycles a bit. A lot of things are connected there for me to go into. So you will meet aspartate again when we talk about the urea cycle. And aspartate, when it contributes to the urea cycle, makes a very important contribution to replenishing your fumarate in your TCA cycle, and that's very important. Fumarate is actually considered, I never said that when we were in the TCA cycle because I have other important things. Fumarate, TCA cycle is about making a lot of energies in the mitochondria. You can trade that NADH, FADH2 very easily. So fumarate going into the TCA cycle would mean about having a lot of energy, contribute towards making a lot of energy. And muscles love that fumarate thing. The aspartate is also precursor in the synthesis of your pyrimidine and purine bases. So in your nucleotide, nitrogenous base, you come across it. Some of it you have to wait, and some of it you already encounter it. Now this is the one that I said we have a common origin that we lost everything and we left one miserable, I shouldn't say one miserable, it's really important miserable pathway, but highly valued. So this is the aromatic. So who are the aromatic? The three: tryptophan, phenylalanine, and tyrosine. If you look at them, they have that aromatic structure. So they're commonly referred to in the aromatic pathway, and they originate. This is PEP. This is phosphoenolpyruvate. So in your glycolytic pathway, when you end up with pyruvate, this is the PEP step. The step right before it is the phosphoenolpyruvate. That one last step is what you do to end up with pyruvate. So this component is part of your glycolytic cycle. So again, from glucose breakdown, you'll get this molecule. Now, erythro's 4-phosphate, you might not remember it because I didn't require you to know that word. It's when you take that side trip around from glucose 6-phosphate. What is this pathway called? The one that you take a side trip and you make NADPH.
Pentose phosphate.
Yes, the pentophosphate pathway, sure. Do you see that? And that's one of the components that you get out of this erythro's 4-phosphate. And these combined with this makes the starting material. So these two are the common origin. It needs both of them. And then you make charismatic. And this is called the Shikinic pathway. But we've lost that. I don't know what happened. Got lazy through evolution. Lost all these enzymes. And then now have to run after the plant to get the materials, right? Basically, we don't have this. So we have to eat plants for this. And where's my stuff? And we have to eat plants, or wait for the plant to be eaten by the cow, and then eat the cow, and you have the whole complete protein. So these two, we have to then get it from outside source now. We can't make them. We can't make this directly either. But we left this pathway to be able to, it's just one enzyme step that helps convert phenylalanine to tyrosine. We kept that. So if we have these in our diet, we're okay. We can convert some of these to that in terms of, so tyrosine is only labeled as non-essential provided you have phenylalanine in your diet. That's what I'm trying to say. Does it make sense? Okay. Now, even though we lost most of the pathway, it's still a very important amino acid. Why? Because tyrosine, which can come from phenylalanine, goes towards making a whole lot of what is called catecholamine. Catechol structure. They all had the same ring structure, and so that's called catecholamine with amine group. And members of these catecholamines are dopamine, epinephrine, and norepinephrine. What do you think they function as? Epinephrine, norepinephrine, huh? And, huh? Neurotransmitter, yes. They are your neurotransmitter. your dopamine. So all these three shares the pathway from, that can either origin from here to or here. Do you see that? So it's very important to have if you want to have good neurotransmitter system. Now, how about tryptophan? Well, it actually helps make your NAD, NADH, NADP. So it makes those. Remember, you need those anyway. You need those to make NADH. Because you can't make NADH just out of nothing. Just like when you make ATP, you need the AMP or ADP to add the phosphate to. So you need this component to start from where? You see? So this has to be made. Right? So of course you can get some of it through, you get some of it from your vitamin, but the vitamin has to be converted too. And they have to combine with something to make them. And so basically your tryptophan is the common precursor to making your NAD, NADP, and as well as it also makes serotonin. And what is serotonin? What is serotonin? Neurotransmitter. It's the one that actually gives you an overall well-being. You will know if you're lacking in serotonin. You won't be here in class. You'll be too depressed, or you have sleeping disorder, eating disorder, mood swing. It's very bad. So you always need serotonin in your system, and it's very important to have it. Now, I don't know whether it's true or not that turkey has a lot of tryptophan. I have to ask the nutrition people that, and that if you eat a lot of turkey, you should be able to make a lot of serotonin. That's linked to, you're always happy at Thanksgiving after a turkey meal. And you're sleepy too after a meal. But I don't know if directly linked to tryptophan, but I read somewhere that, okay, turkey has a lot of tryptophan. But you also know that there's always other system involved. Yeah, you can make a lot of serotonin, but sometimes the cells uptake and degrade it very fast. So your system has to balance up the rapid uptake and the breakdown of it as well. So now you understand why we learned it. And amino acids is not just about protein. It can be building other materials that for your overall health and wellness. Two more families. This is the pyruvate family. And of course the pyruvate family, members of the pyruvate family is alanine. We could guess that because you remember that pyruvate can gain an amino group and become alanine. Valine, leucine, and isoleucine are also part of that family member. And who's the common origin? This time it has the name. Do you see that? So that's your common origin in this case. So pyruvate is your common origin. Anything interesting you have to say? Just like alanine is important for your glucose alanine cycle, right? The other are the branch chain. One more family. This is histidine. So I've actually covered 20 amino acids. So this is histidine. Histidine is a bachelor, don't have any family, looks by himself. Look where he starts out. It's the longest name ever in biochemistry. Phospho That's how long he starts out. No wonder he's a bachelor. So you see, he has to start out there and then you make histidine. So that word sounds very intimidating. Let me show you what that is. Let me draw. That's what it is. Not that scary. So where did it originate? It originates as this molecule here. What is this? What's this molecule? It came out of this.
The phospho-5 ribose.
Yes, it's the ribose 5-phosphate. You see that? This is a ribose and the phosphate at the 5th position. And all I did was add 2 more phosphate at position 1. And then it becomes the phosphoribosyl pyrophosphate. Oops, you can short form it, since it's such a long word. But do know the whole word, because you know what, if I pick an exam question that uses the long word instead of the short form. That's how it is. So, you know, even though it has a long word, it's not such intimidating if you look in and say, oh, it's only your ribose 5-phosphate with 2 additional phosphate attached to it. Do you see what I mean? And so you know it originates from the pentophosphate pathway. That's why we did that pathway too, because you know we couldn't ignore some of the materials that are essential for making, even though this is a bachelor, he's worth quite a bit. Histidine. Now we just make, you know, enough. In adult, we make them enough. In children, they don't have enough. So now you know to study it based on family, name of the family, the common origin, all right? And this is all how they're connected. It's not that scary. It looks scary when I present it like that, and but if you go bit by the time. Let's start with the ones that is easier to see. All right, here. Remember glutamate family? They just start from alpha ketoglutarate, which is one of your TCA cycle intermediate that you already know. It's not something that you didn't know. It's part of your, and we call these, when we use this, then this becomes your anabolic part of that TCA cycle. Most of the time you see the catabolic role of your TCA. Now you see an anabolic role to your TCA cycle. And then of course, since we're in that, your oxaloacetate can be used to make these. Of course, we lost all these, but we have that. The plants still have this, but we have this that we can make out of your TCA cycle again. Another anabolic property of the TCA cycle. And then of course, this is something that you are familiar with. Pyruvate. This can be the origin to pyruvate, which is the end product of glycolysis. Goes towards making your branch chain, makes alanine, makes your branch chain amino acid. And that's your pyruvate family. So you have the glutamate family, the aspartate family, your pyruvate family. And then I told you that serine came out of the glycerate 3-phosphate, which is after the break of your glycolytic part of your glucose into 2-3-carbon component, and then this now goes towards making your serine family. And then of course I have to pull down the chart a little bit to see other members. So basically you're building out of your glycolytic pathway, your phosphoanol pyruvate can be linked together with erythrospor phosphate. It comes out of your pentophosphate pathway to make your aromatic. We lost this, of course, but remember, you can still connect the phenylalanine to tyrosine, which is not on this chart. So this is your aromatic family. And then, of course, out of the pentophosphate pathway, you have your ribose 5-phosphate that makes your phosphoribosol pyrophosphate, which goes towards making your histidine family. So you can see how, so your sugar pathway, now you see so much use for it. Before you were only seeing one direction, mostly the breakdown to create energy, and then the building back of glucose with that. Now you can see that it's a lot more than what it seems. Not much time. Let me see. What is the next? I can read the next one. We are at 11 o'clock, right? Okay. You can stop here. I'll be happy if those